It's great to be here. My name is Anticevic. I'm an associate professor of psychiatry and neuroscience at Yale, and I'm very excited to talk to you about Manifest. We're still a stealth company. We're post-seed, so you won't find much about us, but we'd be happy to talk to you more as I go through this deck. So our mission is to transform CNS therapeutics through precision computational neuroimaging. So I want you to imagine a future in which every person who has a central nervous system disease can get neuro-informed, precise drug targeting. And so we're here to solve that problem. So this is our standard disclaimer. We've assembled a team of international leaders in computational psychiatry and neuroimaging. I'm joined by my co-founders, Marcus Helmer, who's the CTO in our company, and my co-founder, John Murray. We have a deep bench of advisors who are leaders in this sector, as well as people who are experts in computational neuroimaging. So CNS drug development is tremendously risky and really expensive. So I want you to focus on this number in the middle. 4% of drugs go from pre-selection all the way to market. The reasons why these drugs fail in CNS is because fundamentally we have an inability to select the right person for the right drug. So if we double click on this problem, you have a cascade of issues that's driving cost and efficiency such that trials fail too late. Lack of prioritization of agents, given poor animal to human mapping, low confidence of mapping neural targets to actual symptoms, suboptimal selection of people who can actually benefit from the drug. And this creates the biggest failure in the transition from phase 2 to 3, which is a huge driver of cost and inefficiency. And that's how you get to that cumulative 4%. So how do we change this? So we're solving key cost and efficiency barriers in CNS clinical trials through a proprietary platform spun out of the co-founders' laboratories here at Yale that addresses 3 questions: developing agents with quantitative animal to human neural target mapping, confirming those neural targets in humans in a way that's quantitatively linked to symptoms, and then finally enriching with the same data objects patient populations that are maximally responsive to drugs. And I'll show you a demo teaser for how this works. So our platform automates multimodal animal and human imaging and analytics to address these key bottlenecks. So if you do this, you fundamentally transform the probability of an asset making it to market, and if you boost the probability of success in the transition from phase 2 to 3 by 50% in just 1 year, that's $1 billion, up to $1 billion of value per asset. And there's hundreds of these out there that are shelved, and this is a massive market. So this technology is built on 10 years of innovation in my laboratory and my co-founder's laboratory. We started in 2012 when an NIH Director's Award awarded to my group that incubated the tech. We had a number of successful commercial collaborations that gave us the signals that we're ready to go. We had a patent awarded that's part of our tech transfer estate in the company, and we have proprietary IP that we're building on. And most recently, Yale side, we've deployed a clinical trial that uses the technology where we have the first ever primary brain-based readout, again, primary brain-based readout for a drug in early course schizophrenia, and that's public domain information. So here is how this works. So we can fundamentally alter the success of a clinical trial. I'm showing you a status quo situation where this is a failed depression trial. The effect size here for sertraline versus placebo was 0.17. That's commercially not viable. That's a failure. It would have required 411 people to succeed. So through our platform, what we're actually capable of doing is we can take neuroimaging data, we can optimize alignment against existing a priori targets from people's neuroimaging data in relation to FDA approved clinical questionnaires and create a selection procedure that allows us to enrich a trial using questions only, not imaging, but imaging is the decision support tool. With this, the effect size goes up to 0.61, and this would have required 35 people. This is our first proof of concept, and we're working on more. We would be super excited to talk to you. Marcus is also here in the crowd, my co-founder, and so looking forward to questions.